Good day, I'm Dr. T, and welcome to my office. So let's say you're an organism, which, I mean, you kind of are, and you want to control your enzymes, but you don't want to control them the same way, depending on where they are. You want some spatial control over your enzymes. Well, in that case, you've got a couple of options. One of the first is an isozyme. This is a pair or more of enzymes. These are multiple enzymes that are effectively the same enzyme, but they are slightly different. And typically, one of the things that's going to make them slightly different is they're going to have different regulatory control. There are other options for them being slightly different, but a classic one is different responses to different regulatory molecules. For instance, if you look at your muscles and your liver, during high exercise, especially once the adrenaline starts get pumping and you know that bear is right behind you and you better be moving it, they need to be doing two different things. Your muscles need to be consuming energy like crazy. Your liver needs to be dumping sugar into your bloodstream to get it to the muscles so once again they can consume it like crazy. Now, for both organs, you're going to want to activate those enzymes to take glycogen and convert it into glucose. So, once epinephrine hits the cells for both the muscle and the liver, you're going to get a nice enzyme cascade. It's going to amplify the signal, and it's going to tell the glycogenase, probably just butchered that pronunciation, to start breaking apart the glycogen and pulling it apart. But here's a slight difference. In your liver, you don't want glycolysis going up. You want it actually to stop. You want it to move over to gluconeogenesis. Whereas in your muscles, they can't do gluconeogenesis to start with, and if they could, they really don't want to do that now. They want to take all of that energy and move that into moving them so you can get away from the aforementioned bear. So, how do you do that? Some of the regulatory enzymes in the glycolysis pathway and in the pathways that regulate those are going to be slightly different between the liver and the muscles. These are isozymes, and because the cell knows, hey, I'm a liver cell, I'm going to turn on my liver isozymes and not my muscle isozymes, even though the genetic code encodes for both for all cells. Now, those in the muscles are effectively lacking a receptor. They are ignoring that, and they're just going straight for energy consumption, where the ones in the liver are responding to the signal with the adrenaline and shutting down glycolysis as much as possible and cranking up gluconeogenesis. Obviously, this is a short-term kind of thing. Eventually, you're either away from the bear or you're bear food. But yeah, so you've got an approach of isozymes. And we have a bunch of different isozymes for a lot of different approaches. This is a very common approach for regulating things differently in different tissues because different tissues are different. Now, what about, let's say I want to make a enzyme. I want to make it one place, but I don't want it to be active there. This would be something like chymotrypsin, which is a digestive enzyme. It breaks it down proteins. Well, I don't want to break down proteins when I'm in the cell. I want to break them down once I'm in the digestive tract. So the way this one works is we create a zygomorph, or a proenzyme. In this case, what we're going to do is we make an enzyme, but it has bits stuck onto it. When it's manufactured, it has too many amino acids. Some of them need to be chopped off, but they're not chopped off just yet. They're shipped to where they want to go, and then an enzyme there chops off the bits that you want to chop off, and that's how you make chymotrypsin work in your digestive tract. Another approach you can do is, remember, proteins are very pH sensitive. Different regions of your body are going to have different pHs. So if you want to have an enzyme that's functional in the stomach, it needs to be functional at around a pH of 2. Hey, most cells, the innards aren't pH of 2, so this is a great way of controlling that particular enzyme. If it's in the wrong spot, the pH will probably be off, and therefore it'll be less or non-functional at all. Now, you actually have another interesting approach with those proenzymes. They can be done, instead of doing for spatial separation, they can be done for temporal separation. Let's say I want to make a bunch of protein, whether it be an enzyme or a protein that's not actually an enzyme. I'm going to need it. And when I need it, I need a lot of it quick. So the way you do that is you stockpile. But sometimes you want to stockpile, but you don't want it to activate. You know, if you've got, you're military and you got a stockpile of missiles, you don't want them randomly shooting off in your stockpile. So you want some way of having like arming pins that you can pull out right before you put them on the plane and go shoot something. Well, proenzymes are basically the same approach. If you think about blood clotting in a bad time is a really bad thing. So you don't want it to do so. But you don't want the blood to clot, or you don't want the blood not to clot when you need it. That would also be very bad. So what your cells do, specifically your platelets, is they store a lot of the proteins that they're going to need for the blood clotting, and actually the proteins that are going to form the clot, in a proenzyme or a pre-modified form. They get clipped. This is actually part of the process where vitamin K is involved, and previously discussed with warfarin. Those proteins are going to get clipped apart, then they self-assemble, or they get some help, but they assemble into the clot. Bada boom, bada bang. If you don't snip off the tails, then they don't form a clot. And all you have to do is start snipping off a couple of bits, and then they'll form the clot. You don't have to go through the whole process of making the things. And thus, yay, you've got some nice, fast-responding proteins for when you need proteins. So with that said, this is a wrap for this particular video, but I'll see you on the next one.